A very pleasant morning, children. Welcome back to our next session. We were discussing about artificial vegetative propagation. Under that, we were discussing grafting. Now, what is grafting? It involves the two individual plants having some desirable qualities. In this, what we do, we take a lower part of the plant, which is referred as stalk. It should have desirable qualities such as resistance to diseases, and that is from bacterial or viral infection, while the upper part is called as cichon. It should be have desirable qualities that it should be bear flowers and fruits. If you are taking orchard plant or a flowering plant. Now, what happens? The upper part of the trunk and the branches are being cut off, and in they are a cut is made in such a position that the cichon can be grafted on to the stalk, and the part where they are attached or grafted on, that is tied with a suitable you can say cloth or a poly bag, and this part is covered with moist soil or fungus. After few days, you'll find that adventitious roots coming out from the grafted part, and a tiny twig of a branch sprouts which with bearing a tiny leaf or bud. This will develop into a new branch bearing the particular flower or fruit. Got it? Then you can detach it, or it can grow as on the same plant. Next, you have got budding. Budding, in this case, what happens? The lower part of the stalk is taken of any suitable stem, and a cut is made here. Got it? And the bud of the suitable plant, desirable plant is taken and inserted it. Then it is covered with the moist soil or moist moss and tied. After that, you'll find that it a small tiny roots coming out, and again a tiny branch coming twig coming out, and then bearing leaves, and ultimately it will grow into the desired plant. So this method of grafting, many new varieties has been possible. Now you have seen that in certain flowers like roses, you find different varieties. You have seen roses with yellow petals, and the petals having sprinkled with yellow red dots. That is all because possible because of grafting. Grafting, budding, and the next most important method is tissue culture, also known as micro propagation. Why it is called as micro propagation? Because in this method of vegetative propagation, what we are taking? We are taking cells, that is the tissue of a leaf or a root or a stem, that is stem tip, leaf tip. Leaf is an organ, stem is also an organ. In this case, what happens? Suppose this is the leaf. Cut it. Now what happens? A small cut is being made. I can show you the diagram here. Just in this case, see what happens. A leaf is taken and a small insertion or cut is being made. Got it? Then it is allowed to grow in a nutrient medium. Nutrient medium means what? That cut small piece of leaf, that at this part also it can be cut. They are grown in a solution containing nutrients and hormones. Got it? What happens? This forms an unorganized mass of cells called as callus. Then again it is transferred to another that is a beaker or a petri dish, where again that unorganized mass of cells is grown in a nutrient medium and you get tiny plants. Got it? This do not require any soil. They all require nutrient solution. Clear? And after that, these plantlets are allowed to grow in the nursery and then they are planted in the main field. And within a short period, you can get different, huge variety or of plants which will take time when you grow it from a seed. So this tissue culture or micro propagation is very beneficial for the farmers or gardeners because within a short period we get different types of good yield of plants and which are free from bacterial or viral infections. Why? Because their initial stage they are grown in the laboratory medium in a sterilized condition. Clear? So you have to remember the different stages: callus stage, plantlet stage, and maturity stage. So what is callus stage? A part of the leaf or a stem or the root tip is taken and allowed to grow in a nutrient medium. Nutrient medium means what? A solution containing nutrients and hormones such as auxin, cytokinin, and nutrients in the form of coconut milk. Clear? And in that case what happens? These mature cells or tissues forms an unorganized mass of cells called as callus. Again they are transferred to another petri dish. They are grown and the nutrient medium are added to it. They form tiny plantlets. Tiny plantlets means what? That is small tiny plants, and then they are allowed to grow in a nursery and then they are transferred to the main field. Clear? So what is the advantage of tissue culture? It gives a good yield and the plants are free from bacterial and viral infection. So you remember all these: callus stage, plantlet stage, maturity stage. So you may be asked in detail also what are the steps in tissue culture. Clear? And what are the advantage? I have already told you. The advantage of tissue culture, the plant, the yield or the plants which we get, they are free from viral or bacterial infection. They provide a good yield, and moreover you can say add one more point that the plantlets or the plants, new plants are formed within a very short period. Clear? So next class we shall discuss about the advantage and disadvantage of vegetative propagation. So that's all for this session. Have a good day. That's all. Thank you.